everyone. Thanks for your time today. My name is Vevian Zhang, and I'm the Senior Product Manager at GenScript. Today it's my pleasure to introduce to you the Streamlined Analytical and QC test with ELISA to accelerate oncology drug discovery and approval. So quickly about the content that we're going to go through today, I'll give a quick introduction on the ELISA kits overview that we provide at GenScript, and then follow up with the deeper discussion on the viral vector titration, impurity test, as well as the PK and ADA ELISA kits. So in case you're not very familiar with GenScript, we already have more than 20 years of R&D and production experience since 2004. And now we offer more than 3,000 proteins, more than 4,000 antibodies, and more than 100 kits product. With that, we're able to provide up to kilogram per year production capacity for proteins and antibody products. With our proprietary technology platform, we are able to, we're the leading providers of mature protein and antibody labeling and conjugation technologies like biotin and HRP. And with our sophisticated kits development platforms, we are now able to provide kits for diverse applications, including physical chemical analysis and functional activities using ELISA, FACS, IVD, etc. And the kits that we provide and the platform that we have developed meet the international quality standards and compliant with ISO 9001 and ISO 13485 for the GMP. And we have state-of-the-art GMP facility that we operate and manage by ourselves, and we can provide the traceable documentations for your regulatory application needs, including IND, BLA, and NDA. So more to our ELISA kits capabilities, leveraging our reagent capabilities that touch base on the last page. We started to offer ELISA kits products back in the pandemic time with our CPAS neutralization antibody detection kit that are approved by FDA under the EUA application, which was the first neutralization antibody detection kit globally that helped a lot during the pandemic time. And since then, we started to expand our portfolio by providing more and more products with the similar technology and our capabilities, including the drug metabolism assay kits for PK and the ADA analysis, the pharmacokinetics and the anti-drug antibody immunogenicity analysis. And later on, we started to provide the viral vector titration for cell and gene therapies, including the lentiviral titer P24 kits, the AAV titer ELISA kits, as well as the mu-LV titer P30 detection kits, and more coming. And we also started to provide impurity detection kits for GCT and the antibody drug development and QC, including the BSA, Protein A, HISTAC, Cas9, as well as dsRNA ELISA kit. And these are just our recent list, but our list are keep expanding very fast. As you may already know, if you work in the AD and QC environment, ELISA kits are critical components in the analytical development and QC testings. The accuracy and consistency are crucial, but on top of that, the simplicity to use the kits is another important consideration to streamline the time-consuming assay development and accelerate the speed to clinic. So with that, GenScript ELISA kits are developed with innovative technologies from the pilot experiment. Move on to the screening and validation of the key raw materials for the antibodies protein collaborators to validate a wide range of the parameters, including the detection limit, linearity, cross-activity, etc. And once that's all validated, we move on to the process establishment step to define the production steps, reaction system dilution to ensure the best performance to meet our goal. And then after that's all set, the product get to move on to the R&D trial production to verify the product specifications and stability. And then the very last step, and the most important, is the performance evaluation and validation for the LODs, linear reportable range accuracy, precision, and all the most important QC testing parameters to ensure the product can deliver the accuracy, stability, and all the important parameters for the AD and the QC goal. And with that very strong developed platform and capabilities, we are able to provide a very wide range of ELISA kits to simplify and streamline the QC and the AD purpose with these features listed here. All the ELISA kits that we provide are sandwich-based, meaning that it comes with 2 highly selective antibodies to ensure the high specificity and the high accuracy. And also with our very advanced and proprietary antibody development platform, we're able to enable very fast protocol with our ELISA kits. That's all within 2.5 hours. And this becomes very important when you consider that you need to do multiple runs in the day. So with our kits, you're able to do up to 3 runs per day, and this can dramatically increase your productivity, as shown on the right side. And also with our high accuracy antibody capability, we're able to provide a very wide linear range, and this becomes very important for the viral vector titration. As you can imagine, sometimes guessing the dilution ratio can be a pain, but with our wide linear range, you don't need to spend too much hassle on that, and it's much easier for it to use. The kits also provide very broad recognitions, especially for the impurity testings. For example, the Cas9 and the dsRNA, it can detect various different type of variants and still deliver very high accuracy. All the kits can be done, the assay can be done at room temperature for incubation, and this is much easier than the 4-degree. And with that, we can still enable the high accuracy and consistency with very low intra-assay and inter-assay CB. So with that overview, in the next, I would like to dive a little bit deeper into the different offerings we provide here. And the first one I want to touch base on is the viral vector titration of the LVV and AAV. So as you may know, if you work in the viral vector production space, the production itself takes a very long time, and including many different steps and very complex, starting from the whole cell expansion to the transfection, viral vector production, clarification, and a few different steps for the purification before the final packaging and the QC. And throughout that complex process of production, there are multiple steps where the virus need to be titered to ensure the quality of the viral vector for both AAV and the lentivirus. And the physical titer is one of the most popular methods to do this. which include two different methods, either using the qPCR or ELISA. And there are pros and cons of the two methods. The qPCR is to measure the number of the viral genomes, and it is normally considered more accurate quantification of the virus particle because of it measuring the genomes, but it's normally more time consuming and not very easy to speed up. On the other side, the ELISA kits is another commonly used method to quantify the virus titration by using antibodies to detect the specific viral proteins. And normally it is faster method compared to the qPCR. And this is a good advantage when you need to do multiple assays or have multiple different viral particles to do the titration. However, one of the concerns for ELISA kits normally is that it may measure the total amount of the protein, including the free protein as well, and this may impact the accuracy of the assay, and this may become a concern for the QC test. And in the next slide, I'll tell you how we solve this problem with our ELISA kits. So as we just talked about, the free P24 protein in the production could be a potential impurity concern. So with that, we came up with a very innovative upgraded version of the P24 ELISA kit, the Pro version, where you can use the magnetic beads to remove the free P24 protein and significantly improve the accuracy of the ELISA kits. So as you know, to do the ELISA kits to do the ELISA assay for the P24, the virus will need to be lysed, and the P24 protein are normally located inside of the virus. However, during the virus production procedure, there are free P24 outside of the virus as well, which can introduce the additional contamination for the ELISA. So with the P24 Pro kit, where you can use the magnetic beads to first remove the free P24 proteins in the solution, and then do the virus lysis. And this will enable you to significantly increase the accuracy of the detection of the titration of the virus based on the detection of the P24 protein. So here is some data, validation data to show you the result of this innovative P24 ELISA Kits Pro. As you can see, the efficient reduction of the interference from the free P24 can be significantly reduced. In the graph on the left here, as you can see, it's detecting the samples of the lentivirus with some spikes of the P24 free protein, and you can see that with the P24 Pro kit, the accuracy is significantly increased even with the contamination of the free P24 protein. And on the right side, you can see that the accuracy is significantly improved in the condition where there are free P24 protein in the sample compared to the regular P24 ELISA kit. So that's the story of the P24 ELISA and how we improve the accuracy with our innovative technology for the titration. And starting from here, I would like to switch gear a little bit to the AAV ELISA kits for the AAV titration. As you know, the AAV is a very commonly used viral vector for the cell gene therapies, especially for the gene therapy. And the AV comes with different serotypes, 1 to 5, 6, 8, 9 are the common ones. And sometimes the researchers may use the niche or mutant serotypes. So there are many different options. And normally the way to do the titration for the different serotypes is to use different ELISA kits. So as you can imagine, it becomes very tedious to maintain and not very easy to use. So at GenScript, with our strong antibody development platform, we're able to develop the AAV universal antibody that can detect all the serotypes of AAV, and we're able to use that antibody in our further ELISA kits development as well, where we offer the AVX ELISA kits. So with this one, as you can see, no matter which serotype of AV you're detecting, you can use the same ELISA kits for all of them. And this can significantly streamline your process and bring you the flexibility that can be very important for the AD and QC development. With reduced time, labor, and cost, you don't have to store all different type of AAV ELISA kits. You don't have to spend time to develop the assay for the different serotypes, but just to develop use one kit and develop the method once, and then they will be ready to go. So this can significantly increase your speed to the clinic. And here are some data showing you how this works actually. If you can, one of the questions that you may have is how does this actually perform considering detecting all different types of the serotypes? And that's a very good question. So as you can see on the left side, the standard curve of this ELISA kit detecting different serotypes of the AAV is actually pretty good and very consistent across the different serotypes with very wide linear range as well. And on the right side, where we compare the performance of the AAVX ELISA kit versus the single serotype ELISA kit, you can see the performance of the detection is also very consistent, demonstrating that this universal kit can detect, can perform the same quality of the titration as the single serotype ELISA kit. And down here are some more validation data for the ELISA kit itself. As you can see, we are able to show the kit can deliver very high consistency across different samples using different batches of ELISA kits. We're able to deliver very high recovery rate that is always between 80% and 120%. And very wide linear range as well, with the samples with different dilution folds. So here is a case study we would like to share with you using this kit to do the titration of the AAV 2.7 M8. The AAV 2.7 M8 vector is an engineered capsid with a 10 amino acid insertion in the AAV surface variable region, resulting in the alteration of an antigenic region of the AAV2 and the ability to efficiently transduce the retina cells following the intraviral administration. So as you see on the right side, we compared the titration using the AV2 ELISA kit and the AVX ELISA kit, and the result is very consistent across the two different types. And we also compared our ELISA kit with another ELISA kit, which you see that the other providers AV2 ELISA kit actually were not able to successfully detected this mutant variant of the AAV2. So this is a good example where it comes very important for the ELISA kit to be able to detect the variants of the viral vector, not only the natural ones, but also the variant, because you wouldn't want to reselect a different kit when you start to test the different variants of the vector. Okay, so that's about the virus titration. And in the next slides, I would like to further introduce to you about our impurity test, also using the ELISA kits. And the first one is the Cas9. As you know, the Cas9 gene editing become one of the most popular technology nowadays for the cell and gene therapy. And with that, the Cas9 impurity detection is a critical and indispensable step for all the Cas-CRISPR technology enabled cell and gene therapy production. Although there are already different options of the Cas9, the ELISA kit on the market, what we can offer differently and that can be significantly beneficial to you is that the Cas9 ELISA kits from GenScript are able to detect a broad variant of the Cas9, and including the different nuclear localization signals, the NLS. And this is very important because you may not only work with the natural type of the ELISA, but also the variant type, the natural type of the Cas9, but also the variant type of the Cas9 in your platform. And also the ELISA kits we provide is ultra sensitive, that it can detect as low as 0.1 nanogram per mil, and this is significantly lower limit than the other providers. It delivers excellent dilution linearity, and this is not impacted by the dilution ratio of the sample, and it can always deliver very high accuracy. So in the next slide, let's jump into some data where we can see more about the performance. So first, about the broad recognition, as you can see, we actually tested different type of variants of Cas9 for different types here, including different mutants, different variant numbers of the Cas9, and different locations of the nuclear localization signals. And as you can see, the standard curve is very consistent across the different variants, demonstrating that the kit has a very strong capability for the broad recognition of different variants. So you are ready to go no matter which type or variant of the Cas9 that you're working on. And it can deliver very excellent dilution linearity. As you can see, the recovery rate is very consistent from 3 dilution factor to the 200. So this is very good performance. And the recovery rate is also very consistent as well across different samples. And the accuracy is again very consistent using different samples and different batches of the ELISA kit. And the next one I want to introduce is the dsRNA ELISA kit, which with mRNA technology become more and more helpful, more and more popular in nowadays gene and cell therapy development. The dsRNA is another important impurity that need to be tested in the QC for all the mRNA-enabled drugs. So the dsRNA ELISA kits that we developed here comes with a very unique dual J2 antibody technology, and this is the only one in the market. that can provide a dual J2 antibody. And the J2 antibody is the most specific one for the dsRNA. So with our strong platform of antibody development, we're able to provide the J2 antibody, two different types of J2 antibodies, as both the capturing antibody as well as the detection antibody. So this can significantly increase the accuracy of your ELISA for the dsRNA. And the kits provide very wide linear range as low as 0.1 nanogram per mil, faster protocol as we discussed before. And we also provide various standards. We have one standard that is included in the kit, but we are able to provide you different type of standards if you're interested. So here is the data demonstrating the versatile dsRNA detection, where you see that we tested the 3 different modifications of the dsRNA samples coded by the same DNA, and 3 dsRNA samples with different lengths, all analyzed by the ELISA kit. And as you can see, the result is very consistent across the different samples, demonstrating its broad application for the dsRNA impurity testing. And here is more validation data. As you can see, the standard curve is showing very high linear range as low as 0.1, which is the only one that can provide this low detection limitation on the market. And the accuracy is also very high. As you can see, we have detected HQC, MQC, and LQC samples using 3 different batches, and the result is very consistent across them. And here in these tables, it shows you the high sensitivity as well on the left for the ELISA kit, where you can see the limitation of the detection can be as low as 0.1. And on the right side, it shows that there's no hook effect for this ELISA kit, which is an important consideration for the ELISA kit. And last one, I want to touch base on here for the impurity is the BSA ELISA kit, our 2nd generation. The BSA impurity test is very important for the biological drugs, and it's mandated by the regulations for all the drugs that the production process using the BSA. And the BSA ELISA kit provided by GenScript is a 2nd generation sandwich type ELISA, which can enable the very high sensitivity and very high precision. And we also provide a reliable standard that is already calibrated and validated, which is rare to offer in the market. And the kit also comes with ultra stability as well, that is more than 12 months. And here are some, again, some validation data to share with you, where you can see the high recovery of the ELISA kit and the high consistency as well, with a CV within 10%, and again, very wide dilution range up to from 40 to 300 as well. And another important component here is that the specificity of the detection is now impacted by the HSA. As you can see, when we spike in the SAA at different levels, the detection of the BSA is very consistent with even with the HSA background. So this is very important because your drug product during the production, there may be BSA and HSA. Both of them may be used, so it's important to detect the BSA without even in the background with HSA. Okay, so with that, I'd like to move forward to our PK and ADA ELISA kit, which is another important type of ELISA kit that we offer here, and especially for the antibody drug development. So as you can see, the PK and the ADA ELISA kit comes with different types depending on how it is, how the target antibody is detected. And we can offer different substrate for the reading out, HRP or TMB. But important is that no matter which format or which substrate, we are able to validate the assay to enable the protocol as fast as within 2 hours. So with that, you are able to actually do up to 4 assays within 1 day. So this can significantly speed your save your time and speed up your production. So as you may know, the regulations for the PK and ADA testings is very strict. And at GenScript, the kits that we developed are validated according to the SCH, FDA, and NIPA regulations. The accuracy of the kits are evaluated according to the guidance document of the CLSI, EP05A3 to ensure its accuracy and reliability. And so with that, the GenScript, the PK and ADA kits can satisfy the needs of this research from preclinical development all the way through the clinical research. As you can see, here's a quick summary of the comparison of the standards for the research use or the QC use ELISA kits for the PK and ADA analysis. And you can see that for the QC ELISA kit, we have done additional testings to ensure the selectivity, specificity, precision, and the drug resistance. And here are some more validation data for the PK and ADA ELISA kit. As you can see on the left, this is a standard curve for the ELISA kit of the pembrolizumab, the Keytruda, the PD-L1 antibody. So you can see the detection, linear detection range is very high with that can go as low as close to 1 nanogram per ml. So this is a very important tool for all the PD-L1 antibody drug development. On the right side, as you can see, we also tested the selectivity of the ELISA kit, which represents that it can deliver in between 80% to 120% precision, indicated by the dashed line here. And these are some representative data. And for more data, I would highly encourage you to visit our website at genscript.com to learn more about all the validation we have done and how these assays can support your analytical and QC needs. So a quick summary for today, we talk about the viral vector titration ELISA kits with a P24 Pro ELISA that can remove the free P24 protein, and the AAVX ELISA for universal AAV titration. Those two can be important tools for your viral vector titration. We also talk about the impurity test, including the Cas9, dsRNA, and BSA. And we also talk about the PK and ADA ELISA kits. So with that, I would like to stop here and thank you all for your time today. And I would like to welcome any questions if you may have.